Assalamu alaikum, everyone. So in the previous lecture, we started discussing the genetic code, and there are 64 codons encoding the 20 amino acids, 3 stop codons, and 1 start codon. The start codon, this is where the translation starts. The stop codons, these are the codons that signal during translation that will signal the end of translation. So as you can see, the genetic code is redundant. So the genetic code is redundant. Redundant means that we have more than one codon encoding the same amino acid. So as you can see here, 4 codons encoding leucine, 4 codons encoding proline, 4 codons encoding threonine. But notice that the codons that are encoding the same amino acids, they differ in the third nucleotide. As you can see, CC, 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 but we have here UCAG. And arginine, for example, is encoded by 6 codons. We have CG, 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 UCCA, but also AGA and AGG. Okay, so some amino acids are encoded by 2 codons, some by 4, and some by 6, and some by 1. So AUG is a start codon, and it's a codon for the amino acid methionine. Okay, cysteine is encoded by 2, histidine by 2, serine by 4, phenylalanine by 2, leucine by 6, as you can see. Okay. But this is the first characteristic of the genetic code. The genetic code is also non-ambiguous. Redundant مكرر يعني. Not ambiguous means مش عشوائي, not random. So each codon will always specify one amino acid. So basically, the meaning of non-ambiguous means the meaning of ambiguous, non-ambiguous, ambiguous ensures that every time CUU will only encode, every time CUU read by the ribosome, it will encode leucine, it will not encode any other amino acid. That's what is meant by non-ambiguous. All right, so now we've learned about the genetic code. Next, we're gonna look at the different stages of transcription and then translation. So we're gonna learn about the details of transcription, the steps of transcription, and then we're gonna learn about the details of translation. So transcription is the first stage of gene expression. Transcription is catalyzed by an enzyme called RNA polymerase. RNA polymerase is called like this because it will synthesize an RNA molecule using a DNA template. And it's called a polymerase because it will catalyze a polymerization reaction. A polymerization reaction, this is the reaction where a polymer, in this case it's a nucleic acid polymer, will be synthesized. So whenever you see ASE, this means enzyme. I'm sure you already know this. So again, this enzyme is similar to the DNA polymerase, but there are some differences, and we're gonna discuss these differences. But it is used to make a single-stranded RNA molecule from using a DNA template. The RNA polymerase does not need a primer. The RNA polymerase uses the same base pair rules as the DNA, except uracil is used instead of thymine. So let me just quickly make a comparison between the DNA polymerase you've learned about in biology 101 course and the RNA polymerase. In terms of, let's say, other names, we can call the DNA polymerase as DNA dependent. Let me use different colors. Maybe. So we could say other names. We have here DNA dependent DNA polymerase. The RNA polymerase is also called DNA dependent RNA polymerase. So DNA dependent means that this enzyme uses DNA as a template to synthesize DNA. DNA dependent RNA polymerase means that this enzyme uses DNA as a template to synthesize an RNA. Okay. So next, the nucleic acid. The nucleic acid synthesized. Here we have DNA, and it is synthesized in the direction from 5' to 3'. Here we have RNA, which is also synthesized in the same direction. In terms of the required substrate, the required substrate here is DNA, because remember, the synthesis of the DNA is basically could be viewed as a chemical reaction catalyzed by this enzyme. So the required substrates, each enzyme requires a substrate, مواد المتفاعلة, DNA and deoxy ATP, adenosine triphosphate, deoxy TTP, thymidine triphosphate, deoxy, because these are DNA nucleotides. So D stands for deoxy ribonucleic acid. RNA, ribonucleic acid. Deoxy GTP and deoxy CTP. So you've learned about this in Biology 101 course. Here, the template, the substrate is DNA, but also we have adenosine triphosphate, cytidine triphosphate, guanosine triphosphate, and here not thymine, but uridine triphosphate. Okay, so these are RNA nucleotides. Okay, this is in terms of the required substrate. In terms of the requirement for a primer, let me use a different color. Required primer. Do you remember what is a primer? So you learned about primers when you discussed DNA replication. Do you remember what's a primer? أو البادئ in Arabic. So the DNA polymerase requires a primer to start making a new strand of DNA the RNA polymerase does not require a primer to start making a new strand of RNA. Do you remember what's the primer is used for? Come on. ولا حدا. لا حول ولا قوة إلا بالله. AUG هذا الستارت كودون ملوش دخل، هذا له دخل بالترانسليشن مش بالدي ان اي ريبليكيشن. بحكي عن دي ان اي ريبليكيشن. اوكي، ترجعوا تقراوا الدي ان اي ريبليكيشن بتشوفوا شو البرايمر. So remember, DNA replication starts by separating the two DNA strands. Then a short, in DNA replication, a short fragment of RNA called RNA primer will make a double-stranded region of RNA and DNA. After that, the DNA polymerase can come and bind. The DNA polymerase cannot just bind to single-stranded DNA and start making a new strand of DNA. No, it requires a region of double-stranded nucleic acid. Okay, so the RNA primer allows the DNA polymerase. So this is the DNA polymerase to come and bind and start making new strand of DNA. Okay, so this is DNA. This is DNA. Okay, that's the job of the RNA primer, to allow the binding of the DNA polymerase so that the DNA polymerase can start. The DNA polymerase cannot just bind to a single-stranded DNA and start making a newly synthesized strand. Okay, this does not happen. You should know this. Did you learn about DNA replication in Biology 101? Yes or no? سؤال، أخذتوا DNA replication Biology 101 ولا لا؟ نعم ولا لا؟ No? دقيقة شوي، دقيقة. Okay, the last property I'm going to discuss is the proof reading activity. Proofreading activity تصحيح الأخطاء. So during polymerization, the DNA polymerase or the RNA polymerase, they might make mistakes. They might add the wrong nucleotides. DNA polymerase can correct mistakes. So the DNA polymerase has proofreading activity. So yes, while the RNA polymerase does not have this activity. Of course, there are other major differences between both enzymes, but we're not going to discuss them today. All right. So let's have a look at how transcription in general happens, the major steps. There are three major steps of transcription: initiation, step 1, elongation, البدء, elongation, استطالة, termination, اللي هو التوقف. Okay? So during initiation, the promoter, the RNA polymerase will bind to a region on the DNA that is before the gene. So this is the gene. It's part of the gene. This is the coding sequence. So the codons are here. Okay? The codons are here. The triplet code is here. And before the codons, we have a DNA sequence or a region of DNA that is called the promoter. The promoter contains the binding site for the RNA polymerase and other proteins called transcription factors. Okay? And once the RNA polymerase binds, it will start transcription at a place called the start point. And then the RNA polymerase at the start point will unwind the DNA. So the RNA polymerase has a helicase activity. This is another difference between the DNA polymerase and the RNA polymerase. The RNA polymerase has its own helicase activity. Helicase is an enzyme that can separate the two strands of DNA. Okay? In DNA replication, the helicase enzyme will first separate the two DNA strands so that the DNA polymerase can start making new copies or new strands of DNA. The RNA polymerase itself has its own helicase activity, so there will be no other protein that will do this helicase activity. The RNA polymerase itself has this helicase activity. So unwound DNA means the two strands are being separated from each other. Then the RNA polymerase will start building a new RNA transcript or RNA primary transcript using one of the strands as a template. So here in this example, the bottom strand is the template strand of DNA. It could be the top, but here in this example, it's the bottom strand. This is initiation. So initiation involves the formation of what we call the transcription initiation complex. So the transcription initiation complex consists of the RNA polymerase, the promoter, and any other proteins that will help with the binding of the promoter. So we'll talk about this in more details when we discuss initiation. And after that, we have elongation. So the polymerase will start moving downstream or in this direction and making a new copy. So I mentioned the word upstream and downstream. These are relative terms. They indicate the location of things along a piece of DNA. So for example, the promoter is upstream the codons. The codons, we have something called the transcription unit. The transcription unit, this is where the transcription will start and transcription will end. It's called the transcription unit. Transcription unit contains the codons, the DNA codons or the triplet codes also. So the promoter is before the codons. That's why we say that the promoter is upstream, before. The direction of transcription is downstream, which means after the RNA polymerase binds to the promoter, it will move downstream. It will move after the promoter. Okay, so these are relative terms to indicate the location of something with the respect of, let's say, with the respect of another location, or they indicate the direction of movement of proteins along the DNA molecule. And finally, we have termination. This is when the RNA polymerase is detached from the DNA, and now we have a complete mRNA or complete RNA transcript. All right, so let's look at some concepts. We have the promoter, terminator, transcription unit. So the promoter is the region where the RNA polymerase will bind. It's a sequence of DNA that is recognized by the RNA polymerase and other proteins called transcription proteins. We have at the end of the gene a region called terminator, which will tell the RNA polymerase to stop transcription. We have the transcription unit, which contains the DNA sequence that will be transcribed and into mRNA. All right, so let's look into more details about the 3 stages of transcription. First, we're going to discuss the initiation. So initiation requires the binding of transcription factor, okay, to the promoter region. So this is, let's say, this is a piece of DNA, and this region, so we could say that the codons starts from here and continues, of course, codons or coding sequences. And this region upstream the codons, so this is the gene. So this is all basically called the gene, but different, the codons, we have DNA sequences that have the codons, we have DNA sequences that represent the promoter. Now, within the promoter, there is a sequence, a specific sequence called the Tata box. It's called Tata box because it consists of Tata, TA, so they call it Tata box, okay? This Tata box allows the binding of proteins called transcription factors, عوامل نسخ. These transcription factors allows the binding of the RNA polymerase. Now, as you can see, this is RNA polymerase 2, which means there are more than RNA polymerases. So in bacteria, there is only one RNA polymerase. In eukaryotes, there are 5 different polymerases. So 5 RNA polymerases. Polymerase 2 is responsible for making the mRNA. Okay? So the RNA polymerase will bind to the promoter after the binding of the transcription factor. And this is what we call the transcription initiation complex. Transcription initiation complex consists of the promoter region, the transcription factors, the RNA polymerase, okay, all binding at the same promoter region, or basically at the promoter region. And after that, we have a nucleotide here at which transcription will start. This is called transcription start point. So this is transcription start point, or transcription SST, SSP, or transcription start site, or transcription start point. Okay, this is where the first nucleotide here will be used as a template to add the first nucleotide on the RNA transcript. So this is transcription initiation. It requires transcription factors. It requires the formation of transcription initiation complex. And here we have transcription factors. They bind to the Tata box. So as you can see, we have DNA elements are important for the initiation of transcription. These DNA elements are found in the promoter region, like the Tata box. We have other proteins important for the transcription start or transcription initiation, along with the RNA polymerase. The RNA polymerase, once it binds, it will unwind the DNA. It will separate the DNA at the transcription start site like this, so that one of the DNA strands will be used as a template for the synthesis of the mRNA molecule. The next step is elongation. Elongation means istipala, which means the RNA polymerase start moving downstream in this direction, downstream the promoter region, and start polymerizing the RNA transcript, or start synthesizing the RNA transcript. The rate of transcription is 40 nucleotides per second. So the RNA polymerase synthesizes 40 nucleotides per second. As the RNA polymerase is moving downstream, it will keep unwinding the DNA, or separating the two strands, and synthesizing and using one of these strands as a template to synthesize an RNA molecule. Yes, the Tata box is found in eukaryotes, in the promoter of eukaryotes. Now, during elongation, the RNA polymerase does not completely, let's say, if we go back here, it does not completely separate the two strands. So it will only separate the two strands, a region of DNA, the two strands within a region of DNA that is only 10 to 20 nucleotides long. So this region from here to here will be between 10 to 20 nucleotides long. So as the polymerase moves, okay, if the polymerase moves, this region will, the nucleotides, the two strands in this region, after the polymerase moves downstream, they will base pair with each other again. Okay. So the idea here is that the two strands will be separated only as the RNA polymerase will be moving within that specific region. So this specific region of DNA, the two strands are separated because the RNA polymerase is already here. After the RNA polymerase moves to the right or downstream, these two strands will come back together. Okay. termination of transcription after the RNA polymerase let me stop here. Let's take a 5 minutes break and we'll continue. At 12. So far we discussed the details of initiation, elongation. The last stage of transcription is termination. So there are different termination mechanisms in bacteria and eukaryotes. Okay. In bacteria, the polymerase stops transcription at the end of a sequence called terminator, and the mRNA then could be translated without any modification. In eukaryotes, the RNA polymerase will transcribe a polyadenylation signal sequence. So the RNA polymerase will keep transcribing at the end of the gene until it will transcribe a polyadenylation sequence. This polyadenylation sequence signal has the following sequence: A, U, A. Of course, this will be on the mRNA. Okay, so this is the mRNA. Then, after this sequence is found in the mRNA and transcribed by the polymerase, then after this sequence, so 10 or 35 nucleotides after this polyadenylation signal sequence, the RNA polymerase will, the mRNA transcript will be cut from the polymerase, and then the RNA transcript will be released. Okay. So I prepared for you, let me see, a slide that will basically help you understand the 3 stages of transcription. So here we have, let's say, a double-stranded DNA. This is the, let's say, let me label this. So this is just a DNA, it's 5', let's say. Oh, sorry. Come on. What's wrong with it? Five' end. This region, for example, is the from here to here we have the promoter region. And this promoter region contains the data box. This curved arrow indicates the transcription start point or start site. And here we have the polyadenylation signal. Okay. These two are, this is the start codon. These are important for translation. Let me use a different, maybe different color here. This is the start codon, AUG. This is important not for translation, for transcription. And this is the stop codon. This is again important for translation, not transcription. So what will happen is that transcription factors will bind first. These are, okay, now I see what. So here we have transcription factors. They bind to the promoter first and to the data box more specifically. Then what will happen after transcription factor binds, the RNA polymerase will bind. So this is the RNA polymerase. Okay. Now, after the RNA polymerase binds, transcription will start here. And the RNA polymerase will be moving in this direction, downstream, until it will reach the polyadenylation signal sequence that we talked about. Then after, let's say, after 10 to 35 nucleotides, after the polyadenylation sequence is transcribed or passed by the RNA polymerase, maybe the RNA transcript will be cut. Okay. So after the RNA polymerase binds here, now the RNA polymerase is now here, and the RNA polymerase started making the first RNA. It will add the first nucleotide of the RNA transcript. So this is the RNA transcript from the 5 prime. So this is the RNA transcript. Okay. So this is the RNA polymerase. RNA. Again, the RNA polymerase will be moving while it's moving. So as you can see here, this region of DNA, which is about, as I told you before, it's between 10 to 20 nucleotides long. This is like a bubble, زي الفقاعة. This is called transcription bubble. Yes, this is a eukaryotic DNA. So in your book, we discussed eukaryotic DNA transcription. And this bubble will be moving along with the RNA polymerase. Because as the RNA polymerase moves, it will continue separating the two strands of a region between 10 to 20 nucleotides in length. And the region that is already passed by the RNA polymerase will come back together. Okay? And here, the RNA polymerase already synthesized the first nucleotides of the RNA transcript. So let's call this is RNA polymerase, the first RNA polymerase that binds. So it's already here. The RNA transcript is long. What will happen after the RNA polymerase moves, another polymerase can bind and start transcription. So here we have 2 RNA polymerases. One is already ahead of the other. Okay? One just began the synthesis of the RNA transcript. The other already advanced or proceeded in the downstream the gene, and now its RNA transcript is longer. So the first, let's say, the first polymerase. So here we have the first polymerase. This is the same polymerase that bind first to the gene. Okay, so let's call this is polymerase X. This is polymerase X. This is polymerase X. This is polymerase Y, and now Y is here, and now a third polymerase binds holds it. So as you can see, more than one polymerase can start transcribing the same gene at the same time. But this one, just in this figure, just started building the RNA transcript. This one already have a longer RNA transcript because it moved further downstream the gene. Okay, and this one already have a longer RNA polymerase because it's almost finished. And now this RNA polymerase already transcribed the termination signal. So now 20 to 35 nucleotides downstream this signal that is already transcribed, the RNA polymer, some proteins will bind here and they will cut the RNA transcript from the polymerase. So this polymerase, okay, so as you go, so this is maybe, this is time 1, this is time 2, after some time, this is time 3. You have to understand that transcription is a very quick process, so we're talking about seconds. Polymerase X binds here. After some time, it will be here. After some time, it's already almost done. After polymerase X binds, another polymerase will bind, and it will start transcription all over again. So now, and after some time, it's already advanced, and now we have this is like a polymerase in the elongation stage. This is a polymerase in the termination stage. This is a new polymerase in the initiation stage. Okay? So you have here more than one polymerase transcribing the same gene at the same time. Okay? This will increase the rate of transcription. Okay? So this is a snapshot of 3 different polymerases at 3 different stages of, let's say, of transcription. Here we have this polymerase X already in the termination stage, polymerase Y in the elongation stage, and Z is already in the initiation stage, is still in the initiation stage. Okay? After the release, after transcription termination and the release of the RNA transcript from the polymerase, in eukaryotes, inside the nucleus where transcription happens, the RNA transcript or the primary transcript or the pre-mRNA next will be modified by a process called RNA processing. So during RNA processing, both ends of the primary transcript are modified or altered. Okay? So both ends of the pre-mRNA or the primary RNA transcript will be modified. The 5' end will receive a 5' cap. The 3' end will get what we call a poly A tail. Tail means tail. Cap يعني زي القبعة. Now, these two modifications are important. Remember that still this pre-mRNA molecule that we are talking about, this one, is still in the nucleus. This pre is still inside the nucleus. So in order to go from the nucleus into the cytoplasm, first it must be modified by receiving 5' cap and 3' poly A tail. The addition of these two modifications is important because they allow the movement of the mRNA from the nucleus to the cytoplasm. They protect mRNA in the cytoplasm from destruction or from degradation. Okay? And they help with the attachment of the mRNA in the cytoplasm to the ribosome in order for the translation to start. So this is an example of a pre-mRNA or a primary RNA transcript. As you can see, it has a guanosine nucleotide added at its 5' end, and between 50 to 250 adenine nucleotides added to the 3' end, and they will constitute the poly A tail. The mRNA molecule that is transcribed also contains different region. So here we have the start codon, the stop codon. These are important for translation. And we have before the start codon, between the start codon and between the 5' cap, we have the 5' UTR. So UTR stands for untranslated region. So the UTR. And we have another 3' UTR, untranslated region. This is the region that contains the codons that will be translated starting from here to here. Okay? All right. Now, still, after the addition of the 5' cap and the poly A tail, the pre-mRNA will be further processed because not all the codons in the pre-mRNA will be translated. Not all the codons or not all the regions are coding. There are some non-coding regions called introns, and the regions that will be translated are called exons. So here, this is a pre-mRNA inside the nucleus. We have this region is basically the 5' UTR, and this region from here to here, and this region from here to here is the 3' UTR. They are not translated. But notice we have regions here from this region, and this region. They are called exons. So this is exon 1, exon 2, and here we have exon 3. Between them, we have introns. So introns are non-coding regions. They are called introns because they are intervening regions, sequences. Exons are the regions that will be expressed. Exons means expressed. These exons, they contain the codons that will be translated into amino acids. So a process called RNA splicing will remove introns and join exons. So as you can see here, this exon contains codons from 1 to 30. This exon 2 contains codon from 31 to 104. This exon 3 contains codon from 105 to 146. So as you can see, after removing the introns, the exons will be joined together to form. This is the mature mRNA, ready for translation. Okay. So the region from here to here contains 146 codons. So here we have 146 codons, which means this region from here to here contains how many nucleotides, do you think? How many nucleotides are found from here to here? From this region, or from here to here? How many nucleotides? Every codon is made of 3 nucleotides, so you multiply 3 by 146. So we have 146 codons. And if you multiply 3 by 146, how many nucleotides? (438) nucleotides. And T means a nucleotide. Now this region will not be translated. So here we have stop codon as the first codon. And here we have, this is the stop codon. And here we have the start codon, This region will not be translated. There's no codons, but still is part of the mature mRNA. This region will not be translated. That's why it's called 5' UTR. It will not be translated, does not contain codons that will be converted into amino acids or will be used to build amino acids. So this region will help the ribosome synthesize a polypeptide made of how many amino acids? So if this mRNA is translated, a polypeptide will be synthesized. How many amino acids will be in that polypeptide? So after translation, we will have a polypeptide. How many amino acids do you think will be in this polypeptide? How many codons do we have here? 146. So they will encode 146. Yes, 146 amino acids. AA amino acids. So RNA splicing is the process by which non-coding regions in the primer and transcript, which are called introns, are removed. And at the same time, the coding sequences or the DNA or the RNA regions that have the codons, the coding regions, they are joined together, and these are called exons, creating an mRNA with a continuous coding sequence. So here, this pre-mRNA, this primer transcript, does not contain continuous coding sequence, because these coding sequences in exon 1, 2, or 3, they are interrupted by non-coding intronic sequences. Okay. After RNA splicing, we have a continuous coding region. RNA splicing is carried out by a large complex of proteins and enzymes called the spliceosome. So the spliceosome contains proteins and some RNA molecules, and the spliceosome will bind to the primary or the pre-mRNA. Some of these mRNAs are actually, they will behave like enzymes. They will cut the intron and join the exons like this. And the intron that is cut out, it will be degraded eventually. All this happens inside the nucleus. Okay? Okay, we will talk more about the spliceosome, inshallah, tomorrow. Salam.